In previous videos, we've learned so much about sugars, all the different isomers that we can have, and all the different diversity. And as it turns out, that's just the start. There's an enormous amount of diversity yet to go, and we can achieve that through the modification of sugars. So at this point, I'd like to teach you about some of these different modifications of regular sugars that we're going to see throughout the course, and you'll certainly see in other classes as well. Let's start at the top of this page with the idea of amination. And this is pretty simple. It's where we take a sugar, like we have down here, galactose. This is a 6-carbon aldose. It is the C4 epimer of glucose. And it is, by the way, the D form of galactose. And if we take this and take any of the alcohols on this and swap it for an amine, which we've put here, it's fairly straightforward. We call this galactosamine. And because we can do this at any particular position, at the 2-carbon or the 3-carbon or the 4-carbon or the 6-carbon, we have to indicate that we're doing it at a particular position. So this sugar is 2-galactosamine. Now we can extend that a little bit. If we look at this right here, that's the same thing. That is a galactose with a nitrogen amine at the 2 position, except we've now taken it and modified that amine so it's no longer an amine. To name this sugar, we're going to start out with the fact that it's based on galactosamine, and we'll put way out front there 2-galactosamine. But at this point, we've attached an extra group. This is what we call an acetyl group. And you can attach acetyl groups. Remember, that's actually just a little acetic acid. And do you know what kind of linkage we have right there? Do you recognize that? NHC double bond O? That's an amide, kind of like a peptide. And if we attach that acetyl group, we are going to refer to this as a 2N-acetyl galactosamine. And we sometimes just abbreviate this as GAL-NAC for N-acetyl galactosamine. Sugars like this, GAL-NAC or N-acetyl glucosamine, they're often used in your body in places like the joints, and they help make the joints lubricated and more flexible. And that's why sometimes people take supplements of glucosamine or galactosamine to help their joints. So that's amination. Let's move on to the next type. We're going to look at the reduction, and there's 2 types of reduction of sugars. Let's look at this first box and use the example of ribose. This is D-ribose. And in reduction, what we do is take away one of the alcohols and replace it with a hydrogen atom. And this is simple to name. We call it 2-deoxyribose. And that's simply reflecting the fact that we start with a ribose, we remove an oxygen at the 2 position. And so deoxy in that sense means we're removing the oxygen. Now you may know that deoxyribose is the sugar that is used in DNA, and ribose is used in RNA. If you don't know that, don't worry about it. In 2 chapters, we'll be talking all about nucleic acids and how these sugars are involved. There's another type of reduction, though, and that's shown on the right-hand box here, and that's where we change an aldehyde to an alcohol. And when we do that, it's no longer truly a sugar because the normal ratio of atoms is changed, but we still think about it as a modified sugar. And if we take a sugar like this triose glyceraldehyde and make it into an alcohol at the first position, we call it glycerol. Remember, OL is for alcohols. So glycerol is going to show up in our next chapter when we talk about lipids and triacylglycerol fats. Lastly, we have the bottom of the page and a couple of other modifications that we like to talk about, and this is called esterification And there's two types of esterifications we can do, phosphorylation and sulfation, and it's pretty straightforward. We're going to attach a phosphate group to a particular alcohol. Now the alcohol is not there anymore, but it's actually now linked to the phosphate group. This is a very common thing to do. We're going to see it in the last 5 weeks of our course pretty much every day. In this case, I'm using the example where we've attached a phosphate group to a mannose sugar, and we would call it mannose 6-phosphate because in this case it's on the 6th carbon's alcohol. Mannose 6-phosphate, by the way, is interesting in that it's used as a tag for proteins used to send them to the Golgi apparatus. So if you're interested in cell biology, you might see that in other contexts. Over here, we have glucose with a sulfate attached, and in this case, we call it glucose 1-sulfate because the sulfate's attached to the first position. Now here's a question for you. This is a good little practice from stuff we talked about before. Which of these two sugars can mutorotate? And likewise, which are reducing sugars? Let's pause for a second and see if you can answer these two questions. So with regard to mutorotation, remember the rule is that if you have a sugar in a particular anomeric form, and here we see the alcohol of the anomeric carbon is down, compare that to the high-numbered exocyclic here up, this is an alpha sugar. The rule is that if it's uncapped, if nothing's attached to that alcohol, it can relinearize and then recyclize in the opposite form. So the answer is, to this question, which of these two sugars can mutorotate? This can mutorotate. Let's make a note of that. Over here with the glucose 1-sulfate on the right, however, the sulfate is capping the first carbon. It cannot linearize, and therefore it cannot mutorotate. Our second question was, which are reducing sugars? And the answer is basically the same thing. The sugar on the left is a reducing sugar because it has an uncapped anomeric. It can linearize and form a free aldehyde, and the one on the right cannot linearize and form a free aldehyde. So it's not a reducing sugar, and the one on the left is. There's one more little thing we need to say about the naming of this. With the glucose 1-sulfate, because it is in the particular anomeric form alpha and has a sulfate attached to it, it's stuck that way. So the name, we actually have to call it glucose alpha 1-sulfate because in this case it's stuck that way. Over here on the left, mannose 6-phosphate, it can mutorotate, so it's neither alpha or beta, it's just both, and we don't put in any alpha or beta for that. So these are a little bit tricky rules, but they are all rules, and the rules always apply and always work. So that's the end of our modifications.